Thank you so much for the invitation to speak to the Euro-BioImaging Consortium. I'm very inspired by what you all have put together. And for us in Singapore, we're fairly new and really getting our colleagues together to have a coordinated effort, both for imaging in general and for very targeted efforts like in infectious disease. So with that, I just want to start with what we're trying to do in Singapore in my lab at Duke-NUS Medical School. So I started the imaging facility about 8 years ago that I run as a core facility together with my lab and all of our efforts. And so I'm actually a cancer person, but the truth is a lot of our approaches and platforms are amenable to a lot of different diseases. So we want to try to cross-fertilize and create approaches that make the most sense depending on what the needs are for our clinicians and for our researchers. So, okay. So, infectious disease, obviously COVID has proved the point that it's a big problem. Before, I would say it was not so much on the radar of most people. And we know that cancer usually gets most of the funding, but the truth is infectious disease affects substantial amount of the global population. Actually, if you look comparatively to other diseases, it's quite substantial compared to cardiovascular cancer, so on and so forth. So there is a substantial global burden of these diseases. And majority of it, I would say, is tied to antimicrobial resistance. But of course, now we know about viral infectious diseases, so we can't avoid the fact either. And so we really have to think about what are the major threats we have to face. And this is from 2019, that's not so far before, not so long ago. But at that time, the major threats, especially in the US, was with respect to antibiotic resistant bacteria. And so now as we start to think about what's the role of viral infection and treating those diseases, the repertoire of approaches we need to address this is quite substantial. But the truth is, as we start to develop translational imaging approaches, be they drugs or probes, we have to face the truth of development timelines. And we know that COVID hit us very quickly, very rapidly, and we had to get vaccines in record time, something that's never been seen in our lifetimes. So the truth is we need to have approaches that expedite this process. And if you look at how it looks for diagnostic applications in drug development, the timeline looks about the same. And this becomes very troublesome because this is supposed to be ancillary and supportive of what drug development is all about. So how can we streamline this approach? Well, then this takes us to the question of how strong is our data? And I think this is a little disappointing for us, especially as we start to take public funds to do the kind of research we want to do, is that a lot of our data is not reproducible. And it's for a lot of different reasons. And truth be told, it could be because our reagents are not validated, our study design is not appropriate, we've not done careful control and power analysis and data analysis, and further, we've not reported it fully in a way that people can further reproduce our results. So taking all this together, that means we keep on having to repeat what others are doing before us. So I love this notion of what Eric is doing, which is to share, disseminate, make sure our approaches are reproducible, shareable in a way that we can really avoid some of these pitfalls. And so, I think this begs the question, where is our role in imaging? And I think a lot of what we do in the molecular imaging domain is really at the forefront of the preclinical, the animal imaging studies. And the truth is a lot of our studies are poorly designed. We have to consider, for example, sex differences in the way we study our probes and our outcomes, the randomization, so on and so forth. So I really want to implore to everybody to always think about how we are structuring our design of our studies and about the materials that we're using themselves. Are the probes fully validated? Have we done all those other confirmatory studies? What are the gold standards that people in the field will accept? We start to present our imaging data together with that. So now just to talk about the realm of molecular diagnostics, it's a very broad field. And the truth is the diagnostics are at the core of personalized medicine. And it's all about the biomarkers. So what are the biomarkers of health and what are the biomarkers of disease? And actually the truth is applies to all other aspects of life, including forensics and biological warfare. Also important to understand the adverse events of a lot of the materials that we work with. And so looking at toxicology and serology and the metabolomic impacts of our diagnostics becomes very important. But what's interesting to me often is, as an in vivo imaging person, is that often in vivo diagnostic imaging is not included in that. And that's because imaging is usually clumped as a therapeutic. And that's specifically true in the nuclear medicine domain, which is what I'll talk about. So why do I think molecular imaging and in vivo molecular imaging is so important? I think it's because it really bridges that translational gap. And that's really where we see a lot of our discoveries are not able to make that impact in patients. And to make that happen, it's not just about the probe, it's not just about the patients, but the entire pipeline and the entire infrastructure that's required to really meet that gap. And then all of those nodes, sort of as you described, how they're all linked together and communicating with one another to make that happen in a timely fashion. So this is really where I think the power of in vivo imaging comes in, where we go from the very invasive to the very non-invasive, and then use all of that reiteratively to get to address the kind of questions we want to address. So I am a nuclear medicine person, so it's not to take away from the other very powerful and robust imaging technologies that are out there. But, for me as a translational scientist, I'm always looking towards creating approaches that work in our cells, in our animals, and ultimately for our patients. And so for me, that's where the nuclear imaging, the PET and the SPECT, have really the potential to do so because of the quantitative nature of the isotopes that are available, and the fact that we can actually pick and choose different isotopes and potentially translate those to what we call theranostics, those probes that could indeed have therapeutic potential if we swiftly swapped out the isotope. And so the premise is the same as what we do in cancer. It's all about identifying the disease, staging the disease, restaging the disease after treatment, and then understanding what's happening as we start to include different interventions, especially in the context of infectious disease. So really the approaches hold true. And I think the goal we're looking for in infectious disease imaging is, can we do what they're doing in cancer now, which is with our probes, can we shorten trials and drug tracer development in a way that makes most sense and also cuts costs? And I think if we do this collectively, we have a really big chance of changing the paradigm as to how we approach infectious disease development. So my lab, we actually try to approach this also from a fairly bird's eye perspective. So we always look towards the molecular target identification to address that clinical need, which is generally the lack of specific probes to identify the disease biomarkers. We, in certain cases, develop our probes in-house. We then do a lot of probe screening and synthesis. We do all the preclinical validation in cells, tissues, and animals. And we have, what's exciting for us is a setup to do the GMP manufacturing and get those probes into patients to start to really throw these into trials in a more rational approach to, again, ask those questions that are relevant, not to us as imaging people, but I think to the experts, the infectious disease doctors, and those that are actually treating our patients and who really need those tests that make the most sense to address the needs of the day. So really for me as a scientist, it's to understand where things go, how and why they get there, and for how long. So the fundamentals of pharmacology, pharmacokinetics, pharmacodynamics. But ultimately, this is what we want to do for translation, which is to build that data of the preclinical data, the portfolio of data we need to really bring all those innovations to the clinic, and ultimately, truth be told, to the market, which is where we need our industry partners. So this is a great review from Sanjay Jain's lab at Johns Hopkins that really starts to dissect what are the different diagnostic approaches for infectious diseases. He focuses on bacterial infection, which is the need of the day, or was the need of the day stateside, and still remains so. And really to look at the traditional, more invasive methods, which are really about the biopsy, as compared to the non-invasive molecular imaging methods. So, to be able to image our patients from head to toe, or animals from head to toe, to really look at the spatial information, the dynamic information of those different hotspots and lesions that are really representing the infection, and then how we can bring in different treatments to then rapidly detect treatment response. Really, that's the name of the day. Can we rapidly detect those changes that we think are targeted specifically to the biology of whatever microorganism we're looking at. So again, this idea of precision medicine applies in this case, but again, we can't have the lens we're going to image everyone. We want to make this as smart as possible. So again, I look at this more as rapidly trialing and advancing our new treatments that are coming to the forefront. And just to show, you know, you look at these as a pipeline of yes, no kind of answers, and how do we use imaging in each of these steps? Again, you can just swap out bacteria for virus, and the same premise holds true. So again, how can we streamline imaging to understand the pathogenesis of disease with these viruses, but also then to understand how treatments can work to either prolong survival or to mitigate infection and transmission. So I bring this up because sometimes the question comes up, well, how specific does my probe need to be? And really, I think it depends. So the workhorse of nuclear medicine remains FDG. FDG is fairly non-specific. It's actually at the interface of cancer, the fact that our tumor cells need a lot of energy, but also inflammation. So any inflammatory cell will also take up FDG. And this also happens in the context of infection. So it's hard to really tease out what is the source of that signal without intense interrogation biologically. Of course, then the effort comes into how do we develop probes that are specific to that pathogen or to that very specific process that's elicited by that pathogen. So it's not a one size fits all. And, you know, and again, to that whole point of infection, bacterial or virus may require different approaches. And so that's something to always keep in mind. So in my talk, I'm really going to focus on our efforts specifically in imaging flavivirus infection. And this is in collaboration with a leader in flavivirus infection, Subhash Vasudevan, as well as the head of infectious disease imaging and treatment in Singapore General Hospital, Jenny Low, as well as a number of really talented trainees that have pushed this project forward. So for those who don't know about flavivirus infection, actually belongs to a very large family of mosquito-borne illnesses. And so dengue is actually 4 different types of one type of virus, and then actually Zika virus is another type. And we may recall Zika from when the Olympics hit in Rio, and all of a sudden there was this fear of children being born with microcephaly because it was present in men's sperm, for example, and could also infect pregnant women. But Japanese encephalitis virus and yellow fever virus also belong to this class of viruses. So the truth is, this is a significant problem, global problem. It infects half of the world's population. And you can see from this picture over here that this is often considered a problem of the global south. But the truth is, as we know with COVID, that there's no such thing as that. It's a global problem. And you still, especially with global warming, you have rising temperatures and more mosquitoes coming up into what was considered more privileged areas, if you will. So this is becoming a significant problem, and certainly has become a major problem here in Singapore, with us having a resurgence, if you will. And with resurgence of infections comes the potential for re-emergences of infections. So what may have passed, like Zika virus, may indeed come back again, since they are harbored in different parts of the world. And again, they're still present within certain mosquito populations. So the map remains the same. And again, with there being a lot of travel, we know that these viruses will infect everyone from all parts of the world. So this is indeed a global problem, and a problem that affects a significant portion of the world's population. So the challenge for us is, truth be told, there's no therapeutics available. And part of the reason why there are no treatments available is that understanding the pathogenesis of these diseases is very difficult. First of all, these diseases present very rapidly. We can't just easily get a human tissue specimen. It's not like cancer where it's like, oh, there's a hot spot, let me take the specimen. We can't quite do that for these infectious diseases. And it sounds a little murky when I say only humans exhibit the clinical disease, but it's to say that a lot of our animal models don't fully recapitulate human disease. So how do we make sure our models make the most sense? And truth be told, there are no strong clinical biomarkers of infection, which means there's no strong clinical biomarkers of treatment response. So we really hope that imaging can serve that role as the biomarker that allows us to validate those beneficial interventions for our patients. So what are the kind of targets that we can go for imaging? Well, we have to understand how these viruses infect, and then what are the downstream events that happen. So when it comes to virus infection, one of the first things that happens, of course, is the innate immune response. So that's sort of our early response indicators, our early potential therapeutic indicators when we talk about interventions. Of course, what may happen downstream could include vascular permeability and other types of cellular activation that can be very critical when we're talking about treating the disease. So understanding where we are in that disease time course will dictate what's the best treatment to get the outcome we're looking for. So again, the imaging biomarkers could be direct, meaning we're looking at the virus itself, indirect in that we're looking at, for example, the downstream inflammatory markers released from these infected cells, or a surrogate marker of outcome or survival or whatever benefit we're trying to measure. So we first start with the lowest hanging fruit, which is actually FDG. FDG PET is our classic inflammation imaging biomarker that we use in cancer day in and day out. Truth be told, we can also use it in infection. And so we really wanted to understand, could FDG PET be used to non-invasively identify sites of active inflammation in our mouse models, and then also monitor drug response during acute dengue infection. And we've actually published on this quite a bit, and so I don't want to go through this too much, but just to show that in mouse models of dengue, as well as using clinical isolates of dengue, we're able to monitor the longitudinal progression of the disease, as well as the treatment response. And then we also took those findings, we brought that into patients in Singapore. So it's a little different from a cancer trial where we first imaged the patient during peak fever. And so they're tested positive for dengue. So we did a serum test to make sure they're dengue positive. And what we see is extremely high uptake in all the lymphatic tissues, all the lymph nodes and the spleen. Of course, the brain lights up. But it's fascinating to see that after they've recovered, about 30 days later, that signal is completely gone. But we didn't see the kind of imaging signatures we identified in the mouse. So again, mouse does not equal human, but this is still very informative because it is lending to an innate immune response that's happening post-infection. We then took these findings and we recently reported them in looking at FDG both in Zika compared to dengue. And truth be told, even though they're the same family of flaviviruses, the presentation of these infections and the response to that infection is very different when using FDG as a probe. So in Zika, we see very similar to what we saw in humans. It is almost primarily a lymphatic marker. So we see the lymph nodes and the spleen light up quite substantially. And actually, it also tracks with the kinetics of the disease. I'm going to go through that a little bit more. So FDG is all great, but it's very non-specific. So now the question is, can we image with a more specific probe of immune response? And so in this case, we selected the TSPO radioligand FEPA. So TSPO has been reported to be upregulated in myeloid cells, macrophages specifically, or glial cells. And we want to understand, could that be used to look at the body uptake and the body's changes of TSPO positive cells following both Zika and dengue disease, and could we use that to monitor treatment response? And so we used the same models we published before. So this is a mouse model that's interferon receptor alpha beta gamma deficient, and then we used both a mouse adapted strain for dengue and a clinical isolate for Zika. This is the Brazil strain for Zika. And just to show you that the mice have very different survival outcomes. So for dengue, they succumb to disease within 4 days, for Zika within 8 days. And you see quite dramatic weight changes that matches with their survival outcomes. And so when we start to look and dissect the timelines for disease, we can call mid-disease day 4 for Zika and late disease at day 8 for Zika, but it's a little earlier for dengue because they do succumb so much earlier. And that's important because if we try to understand the biology of the disease and the treatment outcomes, we need to make sure we're using the right time points in these very acute dynamic diseases. So just to quickly go through the method. So we do tissue biodistribution with our trace that we injected IV. We do immunofluorescence imaging to confirm changes within the tissue with respect to TSPO itself. So again, that gold standard validation. We do autoradiography with these tissues ex vivo to show in a lot more detail exactly what is the distribution within those tissues. And then, of course, the holy grail, which is to allow us to translate some of these findings, is to look at FEPA uptake longitudinally in our mice during the different disease outcomes, and then, of course, interventions. So really interesting findings that we started to pick up on is that there are differences in the FEPA uptake profile between Zika and dengue. And this is actually, just to be clear, at late disease. So the lungs, the liver, and the spleen do show differences in FEPA uptake, and all are higher than the control. And when I say control, these are mock infected animals. And so this was at least for us an indication that there are changes within these tissues that are quite different from what we see in FDG. And I want to draw your attention especially to lungs and liver, where with FDG there was actually no signal whatsoever. And for liver it was quite important because we see active viral replication in the liver, but we saw no FDG uptake. So we don't see that aspect of the inflammatory response when it comes to using FDG as a probe, but we're seeing something very specific for FEPA. So again, if you're trying to understand, are we clearing virus, maybe FEPA will be a lot more sensitive because it's more specific in nature to those macrophages. So as we start to think about, well, what's the treatment we want to include, we started to use a drug that our colleagues are pushing here in Singapore as a drug that's being repurposed from hepatitis B, and it was dropped some years ago. So tons of safety data. And so we've got an optimized treatment regimen in mice. So it's four times daily dose of selgosivir to these mice starting at 2 days post-infection. And then we start to do imaging at different times post-treatment. So just to show that when you give the treatment, 100% benefit. So all these mice survive, whereas they would have died by about day 4, day 5. In this case, they survived longitudinally. We also see there's a substantial decrease in the amount of active viral replication in the blood, not down to 0, but still quite a substantial decline. So again, this drug is doing something to clear out the virus from the body. Now, if we start to look at the virus within the different tissues, we also start to see a dramatic decrease in the amount of active virus replication within these tissues. And so you start to see that at day 4, which is the lighter green bars, and then it starts to go down quite appreciably at the later day 8. But I do also want to highlight that the brain doesn't see that decrease as one would hope. And that could probably allude to the fact that this drug probably doesn't cross the blood-brain barrier. So again, these are all very important considerations as we start to think about the treatments, where they go, how long it takes to get there, so on and so forth. So with this drop in viral load, does that mean the amount of TSPO is changing? And that's what our probe is binding to. So actually, when we start to do immunofluorescence imaging, and this is specifically to the lung, just to show you at day 4 as the baseline, we have very high TSPO expression, and we see that it certainly lines up with the alveolus of the lungs. And then when you start to see with treatment, this is day 4 post-treatment, that signal intensity is going down, and by day 8 post-treatment, you see it's gone down quite substantially. Also, the number of these very small alveoli also decreases, and we have to go do some further quantitation on that. So again, we know at least that TSPO in these tissues is going down. What does that mean for our probe itself? So we started with, I'm just going to reveal all of this. So we did autoradiography, where we just take all the tissue out and we put it in for whole mount exposure to a phosphor screen. And just to show that at pre-dengue, there's no active infection. This is just what the baseline imaging signal of the probe looks like. So the spleen does light up, and then you see the heart and the lungs. And with dengue, you see that the liver lights up quite substantially, even the stomach and a little bit of the small intestine. By day 4, the liver becomes extremely pronounced. And remember, by day 4, those mice have succumbed to disease. With treatment, the signal looks very close to pre-dengue. And even at day 8, you see the signal pretty much stays at baseline. So this was indeed a very encouraging sign to say that there is a change when we look at the tissue as a whole, because sometimes in PET, we don't have that kind of resolution to be able to separate what these different signals look like. So we went to the FEPA PET. Again, this is at baseline and looking at the lungs and the intestines specifically. So the transverses here is for the liver section. So at day 2, we start to see the signal go up. Day 4, even more so. Day 2 is actually earlier than what we saw with FDG. FDG only picks up in signal intensity at Day 3, and that signal comes down substantially at Day 4 and Day 8. And remember, with the treatment, these mice survived indefinitely. And so when we start to do quantification of this in vivo signal, again, we're starting to see this trend towards decrease in signal post-treatment. Of course, this is pilot data. We still have to increase our N, but it's certainly in the right direction. And our BioD supports all of this as well. So this is all unpublished data. We're still trying to really wrap up the story, but just to summarize and conclude some things from this work. So first of all, it's that FTG and FEPA, both are nuclear imaging probes that are routinely used in the clinic, do reveal very specific tissue-specific signatures in infection, particularly for Zika and dengue. And they can be fairly robust proxies of disease severity. So you don't have to cull the mice every single time and break down the mice. We could use these tracers as proxies of disease severity. Second, FEPA does perform a lot better than FDG as an early response biomarker of disease severity. But just to indicate as well that we only tested cellgosivir, the treatment, in the dengue model. And the reason why we did that is because we actually don't have any promising drugs for Zika at this time. So we're actually working with our colleagues to identify potential vaccines, for example, or antibiotic therapeutics that may work for us to validate how these imaging biomarkers may work as predictive indicators. So future directions, it's to show that indeed FEPA can be used to monitor treatment response in Zika. And again, the limitation is what drug. But also to validate, I think that's the name of the day, it's to validate that our probes don't just give us a pretty picture, that the picture translates to the biology, so that there's indeed the FEPA tracks with the changes in TSPO, that it tracks with inflammation, but also tracks with the very specific immune cells that express TSPO as that marker. So what's on the horizon for us as a lab? There's a lot more opportunities for different imaging biomarkers for dengue especially. So one of the things that patients present with is hemorrhagic shock, which ties back to vascular permeability. So how can we image vascular permeability in our mice and in our patients? Can we actually image the virus itself that's actively virally replicating in the blood and in the tissues? And what does that mean for us in trying to discriminate that signal in vivo? As well as looking at immune infiltration. We're in the era of immunotherapy, understanding the different immune cell populations, and with targeted probes to those immune cells becomes very exciting. And then also for Zika, we're hoping to have a paper accepted very soon on how we're using FEPA to dissect neuroinflammation patterns with respect to disease severity over time. Now, to wrap everything up, what's the hope for us in clinical nuclear medicine? This is agnostic of pathology, but the truth is, to improve our patient outcomes, there's a lot of different considerations that have to be on the table. So for us in Singapore, what's available to us? We don't have all the fancy radionuclides. We're just a small little island in the middle of Asia. So we're really limited about what we have access to. We're lucky to have been able to really get GMP chemistry up and going to get these probes into our patients. But we have to make sure we do all the very intense pharmacology, dosimetry, toxicology to ensure that indeed we're not adding additional burden to our infectious disease patients so that they don't get, for example, the cancer, which is always the fear here in Asia. There's a major fear of radiation. And of course, the right clinical partnerships, not just the oncologists in this case, but the infectious disease doctors and the other key stakeholders that are going to drive the translation of these into our patients. Instrumentation matters. These fancy pictures need very fancy instrumentation, all the engineers to support that. And then the partnerships with academia, with our clinicians, our industry partners, and I would say also our government agencies. And I think the Euro-BioImaging consortium is a prime example of that. And I think the regulatory agencies also have to come into play for infectious disease imaging. Cancer is, everyone's been there, done that. It's not such a scary paradigm. When you start to introduce imaging into sick people with infections, a little bit more different. And so that requires then tremendous amounts of education, but indeed very, very possible. So with all of that, I want to say a huge thanks to my team that just have the energy to make this happen. And I have to highlight Carla Bianca, who's actually a virologist who came to my lab post PhD and really took on all these projects with such dedication and energy. And then all of our wonderful team to support. And of course, I can't do this without the experts in infectious disease, which is Prof. Subhash Vasudevan and Jenny Lowe and their respective teams to really help us get these into the patients and to really address the right questions, use the right drugs and the right models. And of course, our funding resources. I wish it could be there in person. I guess we're still back to Zoom, but perhaps one day you guys can come to beautiful Singapore and see the kind of work we're doing here live and in person. So with that, thank you. And I'm happy to take